And thank you, team, for having me give a talk on the aspects of understanding an NGS report. I'm basically a geneticist. I work as the therapy area expert for personalized healthcare at Roche. Today I'll be talking a bit about the nuances to look for in an NGS report, and these are the disclaimers. And the agenda is I will give a very brief introduction to CGP, comprehensive genomic profiling, and NGS. Followed by what are the components of the report, a brief outlook of the report, overview of the report. Then I will deep dive into two particular cases of comprehensive genomic profiling reports and how one can exploit the report to the best possible way so that the maximum information is extracted out of a report. So for this, it might be a bit redundant, but for the sake of covering the basics, I'll just give a very brief about what NGS and CGP is. So essentially, any cancer is caused, the genomic mutations in a cancer can be classified into four major types. One is the common base substitutions, where a single point mutation happens. Say, for example, in this sequence, ATAGC, it becomes a ATCGC. The A has become a C, which leads to change in the protein's amino acid codon. And almost 95-96% of all mutations that we come across in various tumors are belonging to this particular class of mutations called as base substitutions. Best example is the BRAF V600E, and you have so many other point mutations. Then you have the insertions and deletions, where you could have a single base getting inserted or a single or a few base getting deleted. Best examples include the EGFR exon 19 deletion, where up to 20-30 bases can very easily get deleted in the coding region of the gene, and that causes the oncogenesis, that causes the activation of the pathway. Then you have the copy number alterations, where instead of one copy in a particular chromosome, the number of copies in the same chromosome increases. Best examples are your HER2 amplification or your MYC amplification, etc., where the number of copies of the gene in the same chromosome increases. So in a cell, we will have two copies, one in each homologous chromosome. The number of copies can increase to 5, 10, 20, 30 also in certain cases. And lastly, the most complex type of genomic alterations includes rearrangement. So rearrangement is an event where a gene from one chromosome gets cut and pasted near the gene of another chromosome. So essentially, this leads to a fusion of two genes that results in chimera. So rearrangement is basically chromosome getting rearranged between two chromosomes, whereas gene fusion is a result of a rearrangement where you get a gene fusion. For example, EML4-ALK fusion or NTRK gene fusions, NTRK1 fusions, or EWSR1 fusion. So a point to be noted is not all rearrangements actually cause a gene fusion. There could be rearrangements where the gene is cut, but it is not pasted with another gene, so you don't get a gene fusion event happening. Now, next generation sequencing is a technology that can detect all these four classes of genetic aberrations in a very accurate format. So basically what happens in an NGS technology is whenever you have a sample, you extract the nucleic acid from it, you chop the nucleic acid into 150 base fragments, and then you hybridize them with what is called as baits or probes. So these probes are the genes of interest against which are sequences of the genes of interest. So we have, say, 22,000 genes, and I'm interested in sequencing only 100 or 200 genes. So these probes are against those 100 or 200 genes, and you capture those genes and wash away the remaining part of the genome. And you end up sequencing this regions of interest using a sequencing chemistry. Now, I'm not going to go into the details of sequencing chemistry. You have at least 4 or 5 major types of sequencing chemistries, but the most common type of sequencing chemistry is the Illumina chemistry. So this is exactly what happens in the lab aspect of an NGS. But once the data is generated, then comes the real challenge of data analysis, where you have billions of these 150 base fragments which are mapped to the reference genome, and you identify mutations and make clinical and biological sense out of it. So NGS also comes in various flavors, what is called as a hotspot testing and a comprehensive genomic testing. So essentially what happens in a hotspot testing is that say you have the EGFR gene and you test only the exon 19 deletion or L858R or P790M mutation, that becomes hotspot. You don't sequence the rest of the parts of the exons, or you don't sequence the rest of the parts of the gene. Whereas in a comprehensive genomic profiling, you end up sequencing the entirety of the gene. Why this is important? Because as we sequence more populations, we see that hotspot testing alone might not suffice, or might not, it has the potential to miss certain mutations. So that's why the comprehensive genomic profiling is definitely suggested by, definitely suggested to take the complete genomic profile into picture. Then you have a comparison of the various techniques. Say you have a capillary electrophoresis, which is Sanger sequencing, IHC, FISH, PCR, hotspot, and CGP. And you see that base substitution can be captured by almost everything excepting immunohistochemistry and FISH, whereas copy number alterations are very well captured by IHCs and FISH. But it is the comprehensive genomic profiling that might be able to capture the entirety of all four major classes of genomic alteration. So with this background of having understood that comprehensive genomic profiling is the best method to capture all possible genetic aberrations in a tumor sample, it is our practical experience that comprehensive genomic profiling also produces a lot of information in the report, which sometimes is overwhelming to understand, overwhelming to interpret. And in this lecture, in this talk, I really wish that I would be able to convey certain aspects or critical aspects of the report, which one might be able to interpret further. So what are the various components of a report? So first, I will just talk on the overview of the components of the report and use certain case examples to understand how best we can utilize a report. So overall, the report looks like this. You first have the basic patient demographics, what is the disease, and the specimen types and everything. Then comes the major biomarker findings. So in this case, the biomarker is the microsatellite and the tumor mutation burden, which are the biomarkers for information on the immune therapy possibility. Then you have the list of genetic variants identified in the tumor. So for example, in this case, you have the EGFR exon 19 deletion, CDK12 mutation, MTAP loss, etc., and TP53 mutation. Then you have the genes related to the tumor type with no reportable variant. So this is a lung adenocarcinoma. So you don't have reportable. So we know that for lung adenocarcinoma, EGFR, ALK, RET, ROS, BRAF, ERBB2 are quite important. MET are important because they are targetable. So except EGFR, the other genes related to lung adenocarcinoma that are targetable don't have a mutation in this. Now, it also highlights the major therapeutic implication or the clinical implication of the report, where targeted therapies with NCCN categories are identified. Basically, these are the drugs that are available for EGFR exon 19 deletion. So the drug recommendation comes like this, wherein you have two columns. One is the list of all the drugs that are relevant to the patient tumor. So this is in line with the AMP classification, that is the American Molecular Pathologist classification, where tier 1 variants are the variants which have a direct FDA approved drug for the particular tumor type. Now, tier 2 variants are those variants which have a therapeutic significance in other cancer types. For example, anti-EGFR antibodies such as cetuximab are implicated not in lung cancer, but in colorectal cancer. So in that case, it becomes tier 2 variant, which is the therapies with clinical relevance in other tumor type other than the one that the patient has. Then you also have genetic variants identified in the tumor with no approved therapy. For example, CDKN2A or B does not have a approved therapy currently available for the patient. Now, it goes on to talk in detail about the type of variant. So here I request you to kindly have a note into the variants, the details given here. Most importantly, apart from the alteration, which we have already seen as exon 19 deletion, there is also something called as a variant allele frequency or the variant allele fraction. Now, this is a very, very important underutilized clinically relevant information that is there in the report. So in this case, the variant allele frequency is 40.7%, which means that of the all the DNA that we extracted from the tumor specimen, 40.7% of them had the particular exon 19 deletion. Now, while I'm talking about the case, I will show you how best we can utilize this particular value. Then it talks about the description of various studies and clinical trials that have gone before, that have undertaken, that have been done for giving therapies against the EGFR exon 19. And you also have the frequencies of the mutation in the particular cancer and the prognosis of the mutation in the cancer. All these are given. Then each drug that can be applied, and along with their supporting data, the clinical trial data, is given individually for the particular report. And lastly, you also have clinical trials that can be that have the particular genetic mutation identified in the patient as an inclusion criteria. For example, in this case, there is a CDK12 splice site variant, which is essentially a variant of CDK12, which is pathogenic in nature. And these are the list of clinical trial, along with the trial number, the trial title, etc., and the location where the clinical trial is happening, which is also given in the report. If at all the patient has an opportunity to get enrolled in this trial, the patient will definitely benefit from it. Now, having seen the various parts of the report, I will deep dive into what are the various molecular insights one can get from a CGP report. So this is a case of a 54-year-old male, non-smoker, and the patient had adenoma carcinoma of the lung, and there was a brain mets and bilateral pulmonary nodules were also there. And initially, the patient tested positive for ROS1 by immunohistochemistry. And before the ROS1 was done, I think the patient underwent whole brain radiotherapy because of the brain mets, and 4 cycles of PEB carbo combo was given. But there was a stable disease, and then the crizotinib was added because of the ROS1 IHC positivity. The disease progressed, and then the patient received Bev plus docetaxel. And now the patient was on maintenance therapy, but the disease was still stable. So the sample was sent for tumor comprehensive genomic profiling in the tumor tissue. Now, this is what is the major finding of the report. What do we see? We see a CDK4 amplification, CTNNB1, that is beta-catenin mutation, APC gene mutation, and you have many genes amplification along with PIK3C2G and a TP53 mutation. Now, the most critical aspect is there is no ROS1 fusion. Now, that could be the reason why the patient did not respond to crizotinib, because ROS1 immunohistochemistries are known to have false positives. So a confirmatory test is very important. Either a FISH or NGS like this is definitely important to know whether the IHC is a true positive or not. Now I'll come to the variant allele fraction story of this. Now you see, the CTNNB1 mutation has a variant allele fraction frequency of 7.9%, APC 18.4, PIK3 18.5, and the 2 TP53 mutations have 26 and 30%. Now, what does this suggest? This clearly suggests there are multiple clones of cells in the tumor with different mutations. For example, the most dominant clone is the TP53 clone, followed by the APC and the PIK3C2G mutation. Now, you see the variant allele fraction of APC and PIK3C2G is almost similar, which means that these 2 mutations could be in the same clone of cell. And there is a new clone that is arising that has almost 8% of beta-catenin gene mutation. Now, how to deep dive into this? Now, overall, if you see this, there are no targetable mutations. That's what one would conclude. But let us try to understand the molecular nature of the tumor. So doing a comprehensive genomic profiling not only tells you what to do, it also tells what not to do. And most importantly, it helps you understand the molecular nature of the tumor. And over time, every tumor's molecular nature changes. So first, let us see what is the role of CTNNB1. Now, number one, where the take-home message here is there might not be any point in having immune checkpoint inhibitor here, because the TMB is 0, MSI is stable, PDL1 was also negative, And most importantly, the beta-catenin gene, that is the CTNNB1 gene, has a mutation. Now, this mutation is known to be showing a negative correlation with the tumor infiltrating lymphocytes. So, CTNNB1 mutated tumors have shown to have resistance to ICI, immune checkpoint inhibitors. Now, moving to the next APC gene mutation, it was a G2610 start, meaning that at the 2610th amino acid, when there was a G that has got deleted. So the protein stops with this position. So this is an inactivation mutation, meaning that this mutation does not activate the protein. This mutation actually controls the expression or the function of the protein. Now, of course, we know the APC gene activates the Wnt signaling pathway. Basically, the APC binds with beta-catenin and degrades the beta-catenin by ubiquitination. So when the APC is not binding with the beta-catenin, the Wnt signaling pathway activates. So what does this mean? Inactivation mutations in APC leads to activation of the Wnt signaling pathway. Now, of course, when there is a mutation in APC, it means the Wnt signaling pathway is activated, and it is also shown in murine models that Wnt signaling pathway also activates the PIK3-AKT mTOR pathway, and mTOR inhibitors are found to reduce the tumor burden in FAP mice models, that is a familial adenomatous polyposis mice model. Of course, this is a lung cancer. It is not indicated, but this is a study that is available in the literature. Now, coming to the beta-catenin mutation, it is an activation mutation. So what does this mean? Even if the mTOR pathway is activated, beta-catenin activation is shown to show resistance to everolimus or other mTOR inhibitors. Now, second one is, which I already told, beta-catenin is also associated with resistance to immune checkpoint inhibitor, and it is seen that it is seen more commonly among female patients, non-smokers, and no significant effect on the relapse-free survival was also observed. This is what we could understand from APC and beta-catenin. The third one was the HER3 amplification.
Sorry for interrupting. Kindly conclude your talk, please.
Sorry?
Kindly conclude your talk, please.
Yeah. So HER3 amplification was also identified, which is associated with poor prognosis and metastasis development. Lastly, the most interesting one in this case was the CDK4 amplification, which is, as we know, CDK4 is the cell cycle protein, which makes the cell, promote the cells to undergo the G1, go through the G1 cycle phase. And in non-small cell lung cancer, CDK4 inhibitors like palbociclib and abemaciclib have been shown certain activity where the median overall survival has improved. So what happened is the clinician after this had got palbociclib, and the patient is now on palbociclib therapy, and the patient is doing fine as of now. So the key takeaways from this report was ROS1 mutation, the ROS1 that was detected by IHC was wrong because NGS did not detect. Immune checkpoint inhibitors are not indicated, and CDK4 amplification promotes a cell cycle through the G1 restriction point, and the CDK4 inhibitors can be tried if the patient is on later lines of therapy. Lastly, I'll just conclude with one case, again, a male non-smoker, the lung cancer case. But what we found interestingly was a BRCA mutation, KRAS mutation, GNAS mutation, and a SF3B1 mutation. Now, BRCA is a splice site variant. Of course, in other tumor types like ovarian or breast, it is indicated for PARP inhibitor therapy. But the variant allele fraction was 48.2. So whenever the variant allele fraction, especially in genes like BRCA, is closer to 50, it is suggestive of a germline origin. And after we saw this, when we checked with the patient did have a very strong family history of cancers. And so further confirmation of the germline nature is being tested. And as we know, whenever a patient has a BRCA mutation, it is sensitive to platinum or PARP inhibitor therapies. Interestingly, the patient also had KRAS and the GNAS variant. Now, studies have shown that whenever a patient has both KRAS and GNAS mutation, it is most probably a mucinous type of carcinoma, adenocarcinoma. And in fact, in this particular case, now the pathology was re-examined, and it was in fact found to be a mixed mucinous type of carcinoma. So what did we understand from this? One, BRCA mutation is in the, could probably a germline, and one can give a platinum or PARP inhibitor. Co-occurrence of GNAS and KRAS mutations are suggestive of invasive mucinous adenocarcinoma. And here comes the hypothesis, which is the actual driver. So is it the BRCA or is it the KRAS? So here is where what we did is, because Foundation 1 also does, the CGP also captures the loss of heterozygosity or the HRD score, we are trying to see whether the HRD is high. If the HRD or the LOH score is high, it means that the BRCA is the driver. Whereas if the HRD is negative, then the BRCA is not the driver, it is a KRAS that is the driver. So it is these kind of information that one can really get from a detailed look into the comprehensive genomic profiling. So with the shortage of time, I had to rush through a few of the cases, and I thank you all for your patience. And I'll be very happy to answer any questions if there are.
Dr. Arun, this was indeed a very, very informative presentation, and really amazed to see how to look in between the lines, and this is truly amazing. So 2-3 questions for you. So when there are multiple mutations in a single tumor, so how do we determine that this is a driver one? So this is sometimes a very, very important question. Secondly, when you find VAF of more than 50%, so it is especially true for BRCA only, or it is true for others also, that there can be a potential germline mutation in that? And then the third question, how do you reclassify the variant of unknown significance? So 3 questions we have for you.
Sure, doctor. So thank you for the questions. I'll answer one by one. Number one is, how do you identify the real driver? So that one can very easily see by, number one, we have to see the tumor type and the mutation that is there with that. So, and the way to classify the driver is to see the variant allele fraction. Suppose, hypothetically, let me say, there is a case of glioma with both BRCA as well as, or some case which has BRCA as well as EGFR mutation. If the BRCA mutation has a variant allele fraction of, say, 75% and EGFR has a VAF of 5%, it is the BRCA that is really driving the tumor here. So the variant allele fraction kind of gives you some idea about which is the driver mutation there. And that also helps you in identifying which targeted therapy to look for or to first give. So if you have EGFR as well as ALK, and you have higher frequency of EGFR mutation, the VAF percentage, you target EGFR first, then the ALK, or or vice versa, depending on the VAF. Now, the second question is about the germline origin. Generally, any variant allele fraction of around 50% plus or minus 5%, I would say, standard deviation, could be suggestive of germline origin. But it's true for all the genes, but generally you don't find germline mutations in EGFR, for example. So it is pertaining more clinically relevant more if it is in genes like BRCA or RAD51 or MMR genes like MSH6 or PMS2, etc. So that's where you have a familial component to it, a germline component to it, and it is worthwhile to investigate further. You can do a simple Sanger sequencing from the blood of the individual for the particular variant. And if that mutation is picked up, it is definitely germline in nature. And the last question on reclassification of the variant, that is a very tricky thing, because any variant classification happens based on the establishment of pathogenesis of the variant. Suppose you pick up a MSH2 variant and it has never been reported in the world. Even if it is clinically very significant, clinically very much correlating, unless you have a cellular study or some kind of a functional study to prove that it is pathogenic, one will not be able to classify it as a pathogenic variant. So one thing to do with the PARP inhibitor story or the HRD story is that the genes of HRR, if they are mutated and if at all it is a VUS, now you have a readily easily available functional assay, which is your HRD score. So if that is actually driving it and you have the HRD score positive, even if the variant classification is VUS, you have, if the HRD score is high, you have a kind of, you can be convinced that you can go with the PARP inhibitor therapy.
So one more question, doctor. So with so many labs coming up and all of them, they are doing now NGS, every other lab is doing that. So how do you rely upon the quality of the lab? How do you see that the report is of good quality? So something to see in the report to tell you that it is a good quality lab we are dealing with?
Absolutely, doctor. So I myself have personally worked with a few labs in India. So number one, you have to look for the accreditation. Accreditation alone does not suffice, but proficiency testing. And then in the report, try to see the depth of sequencing, which they have done. Ideally for somatic, it should be 500x. Anything less than that might reduce the quality. There is a very good possibility of getting a false negative. And most importantly, look for clinical and analytical validation. Anything less than 1,000, 5,000 samples, that are 5,000 samples were used for validation, means that the assay is not very much validated. So that is something very important. So doing just a few hundred samples or of tens of samples and saying that it is validated really makes no clinical or analytical sense to it.
So I think more than 5,000 samples, they are huge size. I don't know many of the labs, they are validating their report with the help of 5,000 samples.
Yes, that is the challenge. And that is why you come across a lot of errors in the report. That is why you come across many a times, wrong reporting, false negatives, false positives in the reports. And that is why it's the point I'm trying to make is it's not a biochemical test where analytical parameter alone is important. It's not an automated one. The real challenge with NGS is that it's not completely automated. You need a human intervention to interpret the data. I sequence my own sample. I'm a normal individual. I sequence my sample. I get 1 lakh variants. How do I make sense out of the 1 lakh variants? Although you have artificial intelligence and other things coming up, there is a human intervention required, and that's where validation plays a very, very crucial role.
Thank you so much, Dr. Arun. So we are getting short in time. It's a pleasure talking to you. Thank you so much.
Thank you, doctor.
Thank you.